A very good afternoon to everyone. How are you all? So today we are going to discuss biological classification. Okay, now let's proceed with the lecture. So classification means is the grouping of organism into certain categories on the basis of their characteristics. Okay, this is what we call classification. And why we are doing classification? To make the study easy. First scientific attempt for classification was done by Aristotle. And Aristotle, he grouped the organism in plants and animals. The basis of the scientific attempt, which was taken by the Aristotle, it was based on the morphology. So the plants, they are grouped into trees, shrubs, and herbs. Animals, they are divided into anima and enema. Anima means where the red blood is absent. Remember this, A. Here A is for absent, okay? Anema, so red blood is absent. Enema, red blood is present. So the first scientific attempt was done by Aristotle. It was based on morphology. Second is the two-kingdom classification, which was given by Linnaeus. And Linnaeus divided the organism into two kingdoms, Plantae and Animalia. So according to Linnaeus, the Plantae, it includes prokaryotes. That means bacteria, include fungi, include algae, bryophytes, pteridophytes, gymno, and angiosperms. Okay, so the Linnaeus, they have kept prokaryotes, which are bacteria, fungi, algae, bryophyte, pteridophyte, gymno, angio, all under the kingdom which is Plantae. And the Animalia, it includes invertebrate, vertebrates, and protozoans. So the first demerit of this classification, or the two kingdom classification, was we know that the bacteria, they are unicellular, right? Protozoans, they are unicellular. So this classification, it couldn't differentiate between the unicellular and the multicellular organism. So this is the first demerit. Second is, we know that the fungi, they are heterotrophic in nature, right? Or non-photosynthetic. And whereas algae, bryophytes, pteridophyte, gymno, angiosperm, they all are photosynthetic in nature. So this classification couldn't differentiate between the photosynthetic and non-photosynthetic organism, because they have kept fungi and algae, bryo, pterido, gymno, and angiosperm in the same kingdom, which is Plantae. Third, we know that bacteria, they are prokaryotes, right? And these fungi, algae, and other organisms, they are all eukaryotes. So the prokaryotes and eukaryotes, they are kept under the same kingdom, that is Plantae. So these are the three demerits of two kingdom classification. Next is the three kingdom classification, which was given by Haeckel. So the three kingdoms are Plantae, Animalia, and Protista. First demerit of this classification is that Plantae still consists of prokaryote, that is bacteria. So this kingdom also cannot differentiate between the prokaryotes and the eukaryotes, because the prokaryotes, they are still placed under the kingdom Plantae. Second, the Plantae kingdom also consists of fungi. Okay, and we know that fungi, they are non-photosynthetic. So the fungi is still placed or still kept in the Plantae kingdom according to three kingdom classification. So these are the two demerits of three kingdom classification. Next one is the four kingdom classification, which was given by Copeland. And the Copeland, he resolved one issue, that is separated the prokaryotes from the eukaryotes because he classified the prokaryotes in a kingdom which is known as MONERA. Okay, now we have Plantae, Animalia, Protista, and MONERA. But the demerit of this kingdom, that fungi is still placed in the kingdom Plantae. Okay. Next one is the five kingdom classification, which is most important and most accepted one. Okay, remember this. So five kingdom classification was given by R.H. Whitaker, and he has resolved the issue that he had kept. He made a separate kingdom, which is fungi. So he has separated the non-photosynthetic organism from photosynthetic organism. So the five kingdom includes MONERA, Protista, Fungi, Plantae, and Animalia. These kingdoms, the classification was based on cell structure, body organization, mode of nutrition, reproduction, phylogenetic relationship. Remember these characteristics. This table is very important. It's given in NCERT. Please remember this table. So the cell type, we know that MONERA, they includes prokaryotes, right? And the sole member of MONERA, they are bacteria. Protista, fungi, Plantae, Animalia, they all are eukaryotic in nature. Cell wall, if we know that in case of bacteria, the cell wall, they are peptidoglycan, right? And peptide, it's a protein, and glycan is for sugar. So the peptidoglycan is made up of protein and sugar. Protista, present in some. The cell wall is not present in all the members of which belong to a kingdom Protista. They're present in some. For example, Luglina. Luglina is a protist, but it does not have a cell wall. Instead of cell wall, they have a protein layer, which is known as pellicle, which provide the flexibility to the Luglina. In fungi, the cell wall is chitin. Okay. And the unit of chitin is nag. Remember this. The Plantae, they have cellulosic cell wall, and in Animalia, the cell wall is absent. Now, nuclear membrane. We know that the prokaryotes, they do not have a well-defined nucleus because the nuclear membrane is absent. But in eukaryotes, they have a well-defined nucleus. They have a nuclear membrane. So, nuclear membrane is absent in MONERA. It is present in Protista, fungi, Plantae, and Animalia. In the body organization, MONERA, we know prokaryotes, they are generally unicellular, right? So they have a cellular organization. Protista, they are unicellular eukaryotes. So they also have cellular organization. Fungi, they are multicellular or loose tissue. Plantae, they have tissue, organ. Animalia, it includes tissue, organ, and organ system. Now the mode of nutrition. The prokaryotes, or the bacteria which belong to a kingdom MONERA, they show a diversity in the mode of nutrition. It could be autotrophic or it could be heterotrophic. If they're autotrophic, it could be chemosynthetic or photosynthetic. Chemosynthetic are those which which uses inorganic substrate as their source of energy. For example, nitrifying bacteria. All the nitrifying bacteria, they are chemosynthetic in nature. They're also known as chemoautotroph because they use inorganic substrate like ammonia, nitrate, nitrite as their source of energy. Right? And their photosynthetic autotrophs are also present. For example, cyanobacteria, the blue-green algae, it is photosynthetic in nature. Now, the Protista, they can be autotrophic or they can be heterotrophic. Fungi, they are heterotrophic in nature. Remember this. Plantae, they are autotrophic because they perform photosynthesis. Animalia, they are heterotrophic. Next, we are going to discuss MONERA kingdom. We know that the sole member of the kingdom MONERA, they are bacteria. And bacteria, they are cosmopolitan in nature. Cosmopolitan means that they're present everywhere, whether it's soil, whether it's air, whether it's water. They're present everywhere. And if you take a handful of soil, you will observe that this handful of soil, it contains hundreds of bacteria, because the bacteria, they are very microscopic in nature. They're very small in size, right? And on the basis of their shape, the bacteria could be of 4 types. It could be cocci, which are spherical in shape, bacillus, which are rod in shape, spirillum, which is spiral in shape, or vibrio, which are comma shaped. Okay. And the bacteria, they are also live in extreme habitat, like Archaebacteria. These are the bacteria which can live in the extreme habitat or the adverse condition, because the cell wall is different, or they have a different composition in the cell wall as compared to the nothing. Mostly the plants, they are photosynthetic in nature. They are known as producers. They perform photosynthesis because they have chlorophyll pigment. Okay, they are mostly photosynthesis, photosynthetic autotroph. Now the bacteria, they are simple in structure. Okay, why they are simple in structure? Because they are unicellular. Because they are unicellular, so they have a simple structure, but they show a complex metabolic diversity. And they also show a diversity in their mode of nutrition. I told you earlier that there can be autotrophic, chemosynthetic, or they can be chemosynthetic autotroph, photosynthetic autotroph. They can be heterotrophic. In heterotrophic, they can be saprophytic, or they can be decomposers as well. Okay, so the bacteria, they are simpler in structure, but they show diversity in the mode of nutrition. Next one is the Archaebacteria. The Archaebacteria are those bacteria which can survive in the harsh condition, or which live in the harsh condition, like extreme condition. Extreme condition could be a salty areas. Those bacteria are known as halophiles. The bacteria which are living in hot springs are known as thermoacidophiles. Those which are living in marshy areas, they are known as methanogens. And why they can survive in these adverse conditions? Because their cell wall structure is different from the eubacteria or the true bacteria. And we know that the methanogens, they are also involved in the production of biogas, the methane gas, which is released in the biogas. It is produced by the methanogens. Methanogens, they are also present in the gut of the cattle, like cows and buffalo. Next is eubacteria. Eu means true. So eubacteria, they are the true bacteria. They have rigid cell wall. And if they're motile, they have flagella. And we know that there's a cyanobacteria. Cyanobacteria, they are also known as blue-green algae. And the example of cyanobacteria are nostoc and anabena. You can see in the image, this is a filamentous blue-green algae, which is a nostoc. And the colonies, they are surrounded by a gelatinous sheath. And the heterocysts are present. Heterocysts, they help in the nitrogen fixation. Remember this. These are the cells which are involved in nitrogen fixation. And the heterocysts, they are thick-walled. Why they are thick-walled? Because the enzymes which are involved in nitrogen fixation, they are oxygen sensitive. Okay, so they are thick-walled so that the oxygen from the environment cannot diffuse into the cell. Otherwise, they will affect the nitrogen fixing enzyme. Okay, so the heterocysts, they are generally thick-walled. And the cyanobacteria, they have a chlorophyll A pigment, okay, which is similar to green plants, and they are photosynthetic autotrophs. Okay, remember this. And we know that the prokaryotes or the bacteria, they does not have a membrane-bound organelles, right? The prokaryotes, they does not have membrane-bound organelles. That means they don't have chloroplast. So where this chlorophyll A pigment is stored, in the cyanobacteria or in bacteria, they have a specialized structure which is known as chromatophore. Okay, so in the chromatophore, these pigment molecules, they are stored. Cyanobacteria, they are unicellular, they are colonial, they can be filamentous, they can be freshwater, marine, or terrestrial algae. Now they are chemosynthetic autotroph. That I already told you, that chemosynthetic autotroph are those bacteria which can synthesize their food By using they can oxidize various inorganic substances like nitrate, nitrite, ammonia, and use them as their source of energy. So these chemosynthetic autotrophs, they're generally nitrifying bacteria, and they play a very important role in nitrogen, phosphorus, iron, and sulfur recycling nutrient. Remember this. In nitrogen cycle, you have studied the nitrification, where you have studied the role of these bacteria like nitrosomonas, nitrococcus, nitrobacter, they all are nitrifying bacteria. We know that the bacteria, the majority of them are heterotrophic. And in heterotrophic, the majority is decomposers. There are good bacteria also, and there are bad bacteria also. Good bacteria are those, for example, which are providing some kind of service or helpful to the environment, like formation of curd from the milk. The bacteria which is involved in this is lactobacillus, and lactobacillus is a good bacteria. Production of antibiotics like streptomyces bacteria, fixing nitrogen in legume plants like rhizobium bacteria, which help in the nitrogen fixation in the roots of the legume plant. And there are bad bacteria also, which are pathogenic to humans being, to crop, to animals, to pets also. Like they can cause cholera, typhoid, tetanus, citrus cancer diseases. And the bacteria, they reproduce by binary fission. Binary means two, fission is division, okay, divide. So from the one parent cell, you will observe two daughter cells will be formed. So the bacteria, they generally divide by binary fission. And in the unfavorable conditions, they are under stressful condition, the bacteria, they can survive by forming the endospore, which is a resting spore. Okay, and when the favorable condition arises, the cell wall of these endospore, they get burst, and the bacteria, and they will give rise to a new bacteria. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacteria to another. We know that the bacteria, they generally show genetic recombination or the sexual recombination through conjugation, through transformation, through transduction. Okay, and we know that all the bacteria, they have a cell wall, right? And the cell wall is made up of peptidoglycan. But there are the bacteria which lack cell wall, and the example is mycoplasma. Remember, this is important. Mycoplasma, it does not have cell wall. Okay, it can survive without oxygen. It pathogenic to plants and animals. Mycoplasma is also known as pleuropneumonia-like organism. Okay, and it also known as joker of the cell. Why? Because it does not have cell wall, so it can acquire any shape or the form. So it is known as joker of the cell. And it is the smallest living organism. Remember this. Next, we are going to discuss Protista. Protista, they are the unicellular eukaryotes. And it includes chrysophytes, dinoflagellates, lugnioids, slime molds, protozoans. They all comes under the Protista. Protista, they are primarily aquatic. And we know that Protista, they are unicellular eukaryote. Eukaryote, that means they have a well-defined nucleus. They will have a membrane-bound organelles. And they can either have flagella or cilia. Like in Luglina, they have two flagella. One short, one long. Like dinoflagellates, they also have a flagella, one transverse, one longitudinal. Paramecium, they have cilia. Okay, and remember the cilia, they are present in more in number as compared to flagella. The cilia, they are present all over the body surface of the bacteria, and they show a coordinated function like a oar. Oar is chappu, which we use to like move the boat. Okay. Protista, they can reproduce asexually and sexually by a process of cell fusion and the zygote formation. First, we are going to discuss is chrysophytes. Chrysophytes include diatoms and the golden algae. Another name of golden algae is desmid. They are mostly found in water as well as in marine environment. They are photosynthetic in nature. Remember this. And diatoms, they are the chief producer. Chief because they are present abundant in nature. They are present in a very large quantity or in abundant in the ocean. Second is producer. Because they perform photosynthesis. So they are known as chief producers. And these diatoms, they have a soapbox pattern. Like their cell wall, it shows a soapbox-like pattern. Like this. It's a box and it's a lid. So This type of pattern is shown by the cell wall of the diatoms. And the cell wall, they have silica. And because of the presence of silica, the cell wall is indestructible. Okay, it cannot be destroyed. So when the diatoms die, these cell wall, it keep on depositing at the ocean bed and they form a diatomaceous earth. It's very important. This characteristic of diatom is very important, that they form a diatomaceous earth. They have silica. Remember this. And they are the chief producer in the ocean. So being gritty, the soil is used in polishing and filtration of the oil and syrup. Okay, remember this. Next one is dinoflagellate. Di means two and flagellates means flagella. So dinoflagellates, they have two flagella. One lies longitudinally and the other is transversely, like this. If this is a So one flagella lies longitudinal and the one flagella lies transverse. And you will observe on the outer side, there's a stiff cellulosic plate are present. Remember this characteristic, that is stiff cellulose plates are present on the outer surface. Okay. And they can be dinoflagellate, they're mostly marine, they're photosynthetic, and they could be of various color. They can show up various color. It could be yellow, green, brown, blue, or red, depending upon the pigment which is present in their cell. So there's a red dinoflagellates. When it undergo rapid multiplication, the sea appear red in color. Okay, and we also call it as red tide. You can see in the image, the sea, it is appearing red in color because of the red dinoflagellates. And when they are produced, when they reproduce in a very large amount, these red dinoflagellate, they also produce a toxin. Okay, and this toxin can causes the death of the other aquatic organisms, such as fishes. Next one is lignioids. Lignioids, the pigments of lignioids are identical those present in the higher plants. Okay, and they're photosynthetic in nature. Lignioids, they have two flagella. One is short, one is long. They can show two type of mode of nutrition. When the sunlight is present, they can be photosynthetic in nature. When the sunlight is absent, then they can heterotrophic in nature by predating on the other small organism. They do not have a cell wall. Remember this, that Lignina, they don't have a cell wall. Instead of cell wall, there's a protein layer is present, which is known as pellicle, which provide flexibility to the body of Lignina. And they're mostly freshwater organism and found in the stagnant water, the water which is not moving. Okay. Next one is slime molds. They are saprophytic. That means they feed on the dead and decaying matter. So when there's a suitable condition, these the cells of the slime molds, they aggregate and they form a plasmodium. And during the unfavorable condition or the stressful condition, these plasmodium differentiate and they form a fruiting bodies which bears these spores. And these spores, they have a true walls. You can see in the image, this yellow color, these are the slime molds. They are growing over the decaying twigs or leaves and engulfing the organic material. Next one is protozoan. Protozoan, remember, they are heterotrophic. They can either be predators or parasite. There are four major groups of protozoan. They are amoeboid protozoan, they are flagellated protozoan, they are ciliated protozoan, and there are sporozoans. Amoeboid protozoans, which includes amoeba, right? And we know that amoeba, they have pseudopodia, which is a false feet, and the pseudopodia, they help in the movement. They also help in catching the prey. They also help in engulfing the food particles. Okay, and the marine form of amoeba, they have silica shells on their surface. Flagellated protozoan, they have flagella, and the example is trypanosoma. And remember that this protozoan, it causes sleeping sickness. Okay, remember this. Next one is ciliated protozoan. The ciliated protozoan example is Paramecium. They have cilia on their surface, on their body surface, and they have a cavity which is known as gullet. And through this cavity, the food is intaken by the organism. So this gullet, it helps in the food injection. Next one is sporozoans. Remember one point regarding the sporozoan, that they have a spore-like stage, or the infectious spores-like stage. Okay, remember this in their life cycle. And the plasmodium, which causes malaria. And plasmodium is a malarial parasite. Next, we are going to discuss is the fungi. Fungi, they are heterotrophic in nature. They could be parasitic, they could be symbiotic, and they can be saprophytic. The fungi, we know that they are symbiotic in nature also. That means they show symbiotic association. They can show symbiotic association with algae also, right? And it is known as lichen. The association is known as lichen. They can also show the symbiotic association with the roots of the higher plants, and the association is known as mycorrhiza. The fungi, they show great diversity in morphology and the habitat. They are multicellular, but the exception is yeast, Saccharomyces, which is a unicellular. Fungi, they are cosmopolitan. That means they are present everywhere. They're present in air, water, soil, on animals and plants as well. Their favorable condition is the warm and humid places. Yes, nursing. You're absolutely correct that lichens, they are pollution indicators, right? And mostly they are sulfur dioxide pollution indicator. They won't grow in the area where the pollution is high. So you won't see the lichen in the polluted area, remember. Okay. Their bodies consist of hypha. Hypha, they are thread-like structure like this. And if they hypha, they form a network, then it is known as mycelium. And the hypha could be septate or aseptate. Septate means that there's a presence of cross wall like this. I'm drawing a septate hypha. These are the nucleus. And this is known as septal pore. This is a septate hypha. But there's a aseptate hypha also present where these cross walls are absent like this. So the hypha, they form a continuous tube. So in aseptate hypha, you will observe the cenocytic condition or the multinucleate condition because there's no cross wall. We know that the cell wall of fungi, it is made up of chitin and polysaccharide. Remember this. Vegetative reproduction could be through fragmentation, fission, or budding. Budding you will observe in case of yeast. Asexual reproduction could be through conidia, sporangiospores, or zoospores. Sexual reproduction is by the oospores, ascospore, or basidiospores. Now let's discuss the sexual cycle, which involves the following three stages. Number one is plasmogamy, which is the fusion of the protoplast of the cell. Next is karyogamy. Karyo means nucleus, so fusion of nucleus will occur. Third is when the nucleus get fused, they form a zygote, and the meiosis will occur to form the haploid spores. So in many fungi, or most of the fungi, when the protoplast of the cell or of the two gametes, they fuses. When the protoplast fusion occur, immediately the karyogamy occur. That is the fusion of the nucleus. And immediately a zygote is formed. But in some fungi, you will observe, like in Ascomycetes and Basidiomycetes, you will observe a dikaryophase. Like Let me draw. These are the two cells. First is the protoplasm is fused. This is a dikaryon stage. Because the karyogamy has not takes place. After some time, the nucleus will fused. Okay, so remember in Ascomycetes as well as in Basidiomycetes, you will observe a dikaryotic phase. And this zygote will undergo meiosis. So in fungi, you will observe zygotic meiosis. And the haploid spores are formed. And these haploid spores, they will form hypha, then hypha multiplies, and they will form, like the hypha, they will form a network which is known as mycelium. First, we are going to discuss is Phycomycetes. They are found in aquatic habitat. On decaying wood, in moist, damp places, or as obligate parasites on plants. Their mycelium is aseptate and cenocytic. Remember this. Okay. And the asexual reproduction is by the zoospore, which are motile. That means they have flagella, or even by aplanospores, which are non-motile. And these spores, they are endogenously produced in the sporangium. Sexual reproduction also takes place by the fusion of the two gametes, and they form a zygospore. The gamete, they could be isogamous, and isogamous or oogamous. Isogamous means iso means same. Okay, so they're similar in structure, and iso means dissimilar in size or structure. And the examples are very important. Okay, remember the example that Rhizopus, the Phycomycetes, it includes albugo, and albugo is the parasitic fungi on mustard. You can see in the image, this white color. These are albugo fungi, okay? Rhizopus is the bread mold. You have observed Rhizopus on the bread. And there's mucor. Remember the example. Example are very important, okay? Ascomycetes, they are all commonly known as sac fungi. They are mostly multicellular, but the exception is yeast. Yeast is unicellular. They are saprophytic. They can be decomposers, parasitic, or coprophilous, which is growing on dung. Mycelium is branched and septate. Remember this. Asexual reproduction is through conidia, which are produced exogenously on the conidiophore. Let's say this is a conidiophore. The first branch is known as ramus. Second branch is known as metula. On the metula, there are stegmata present and the spores are. These are conidia. And these are arranged in a basipetal manner. That is the youngest at the base and oldest at the top. And the sexual reproduction occur through these ascospores. The sexual spores are the ascospores. Their ascus are present, right? There's a fruiting body in Ascomycetes. You will observe ascocarp. Inside the ascocarp, there's a ascus is present, and these ascus, they form the ascospores. Remember the examples. Example of every class of fungi is very important. That Ascomycetes includes Aspergillus, claviceps, Neurospora. Neurospora, they are generally used for the biochemical and the genetic work. Okay, remember Neurospora. Their morels and truffles are present. Remember these, that morels and truffles, they are edible fungi. That means you can eat them. Next one is Basidiomycetes. The common name of Basidiomycetes is bracket fungi, puffballs, club fungi. Okay. They grow in soil, on logs, in tree stumps, in living plant body as parasite. Example, rust and smuts. Mycelium is branched and septate. Remember this. Asexual spores, they are generally not found, and sex organs are also absent in basidiomycetes. So, but the plasmogamy is brought by the fusion of the somatic cells of different stains or genotype. Okay, and then the dikaryotic phases form, which give rise to a fruiting body. So the basidiocarps are present. Inside the basidiocarp, there are basidium are present, and these basidium, they bear the, or they have these basidiospores. Example of basidiomycetes, there are agaricus, which are mushroom, astilago, which causes smut, puccinia, which causes rust in wheat. Remember this, okay? It's important. The examples are very important. Nursing, that I have to check. Okay. Next one is deuteromycetes. Deuteromycetes, they are known as imperfect fungi. Remember, that's very important, okay? That they are known as imperfect fungi. Why they are imperfect fungi? Because the sexual stage is not known, or in case of deuteromycetes, so they are known as imperfect fungi. Their mycelium is septate and branched. Remember this. Remember the example of deuteromycetes, that is Alternaria, Coleotrichum, Trichoderma. Also remember that they help in mineral cycling. These are the important points, okay? Regarding deuteromycetes, please remember these points. Next one is virus. Very, very important topic of this chapter, okay? The virus, viroids, prions, they are very important. So according to Dmitry Ivonsky, he recognized a certain microbes as a causal organism of the mosaic disease of tobacco. So he observed that there's a organism which is, or a microbe which is causing a disease or mosaic disease in tobacco, and he find out that this microbe is even smaller than the bacteria, because this microbe can pass through the bacteria proof filter. Later, there's another scientist, which is M.W. Bijernik. He demonstrated that he extracted he'd taken an extract from the infected plant of tobacco, okay, and when he exposed this extract to the healthy plant, he observed that the healthy plants, they also get infected. So he called this pathogen as virus, and the fluid is known as contagium vivum fluid, which is an infectious living fluid. Remember this. And the crystalline nature of the virus was explained by the W.M. Stanley, okay, that the crystal, they consist largely of proteins. Remember this, okay? And the virus, they are obligate parasite. They are inert when they are present outside the host cell. They are inert. When they enter the host cell, they start using the machinery of the host cell and they start multiplying. The virus, they are nucleoprotein, because they have nucleic acid and they also have protein. So the infectious agent is the genetic material. Remember this, that the genetic material is the infectious. In virus, the protein coat is known as capsid, and the unit of capsid is capsomere. And the genetic material can either be RNA or DNA. In case of viruses which are infected the plant, they have single-stranded RNA as their genetic material. The viruses which are infected the animals, they can either be single-stranded or double-stranded RNA or double-stranded DNA. Remember this. It's important. Viroids, they were discovered by T.O. Denier in 1971. They're smaller than viruses. Remember this, that they are smaller than viruses. They don't have a protein coat. They have RNA as the genetic material, and this RNA has a low molecular weight, and they causes a disease which is a potato spindle tuber disease. Remember this. All the characteristics regarding virus, viroids, and prions, they are very important. I'm giving emphasis on these. Okay, they're very important. You might get a question from these topics. Prions, they are abnormally folded proteins. They cause neurological disorder. They are similar in size to virus. They can cause the BSC disease in cattle, which is a bovine spongiform encephalopathy. And in humans, they can cause CR-Jacob disease, which are the neurological disorders, because they causes abnormal folding of the proteins. Lichens, they are a symbiotic association between algae and fungi. Okay, the fungi, they provide shelter and help in the absorption of the nutrient and provide it to the algae. They absorb the nutrient from the soil and the water and they provide it to the algae. In algae, it is photosynthetic in nature. So it help in providing the food to the fungi. So in both the species, they are getting the benefit. So they are a symbiotic association. Algae is known as phycobiont. And fungi, they are known as mycobiont, okay? And the lichens, they are very good pollution indicator. They do not grow in polluted areas, okay? And especially, they are very good pollution indicator of sulfur dioxide. Here's the image you can see. This is the image of lichen. Now let's proceed with the question. Question number 1 says, identify the fungi which do not belong to the group of other fungi among the following. So you have to find out the odd one out, okay? Option 1 says puffballs. Option 2 says mushroom. Option 3 says bracket fungi. Option 4 says sac fungi. What do you think? What will be the correct option? Identify the fungi which does not belong to the group of other fungi among the following. Very good, Chishni. Puffballs, mushroom, bracket fungi. They all are the alternative name of Basidiomycetes. Sac fungi is the Ascomycetes. So the correct option is fourth. Moving next, which of the following characters belong to the kingdom Protista? 1 prokaryote, 2nd multicellular, 3 all members have cell wall, 4th presence of nuclear membrane. Which of the following characters belong to the kingdom Protista? Nursing, they're asking which of the following characters belong to the kingdom Protista. Very good, Shishni. Question says, which of the following characters belong to kingdom Protista? Protista, they are unicellular first. They are unicellular eukaryotes. Option 1 says prokaryotes. No, they are eukaryotes. Option 2 says multicellular. No, they are unicellular. Option 3 says all members have cell wall. No, all members, only some members, they have cell wall. I've given you the example of Euglena. Euglena does not have a cell wall. They have a protein layer which is known as pellicle. Okay? So not all the members, they have cell wall. Some members, they have cell wall. They are eukaryotes, right? And the eukaryotes, they have a well-defined nucleus. They have a presence of nuclear membrane. So, presence of nuclear membrane. This is the correct option. I hope it is clear to everyone. I hope it is clear to you, nursing. Moving next, which one of the following is wrong for fungi? Option 1, they are heterotrophic. Option 2, they are both unicellular and multicellular. Option 3, they are eukaryotic. Option 4, all fungi possess a purely cellulosic cell wall. They're asking about the wrong, that is incorrect statement. Very good, Veena. Very good, Sushant. Very good, Shishni. Very good, nursing. So the question is asking which one of the following is wrong. That is incorrect for fungi. They are heterotrophic, yes. They are both unicellular and multicellular, yes. Unicellular are yeast Saccharomyces, which belong to class Ascomycetes. They are multicellular also. They are eukaryote. Yes, fungi are eukaryote. All fungi possess purely cellulosic cell wall. No, they have a chitin cell wall. Next one is the imperfect fungi, which are decomposers of litter and help in mineral cycling belongs to. One is Ascomycetes. Second is Deuteromycetes. Third is Basidiomycetes. Four is Phycomycetes. This I've told you. Answer this quickly. Very good, Veena. Very good, nursing. Remember this term, imperfect fungi. Very good, Sushant. Very good, Shishuni. The imperfect fungi, which are decomposers of litter and help in mineral cycling. Remember the mineral cycling and the imperfect fungi, that the characteristics of Deuteromycetes. Why they are called imperfect fungi? Because sexual stage is not known. They only reproduce by asexually, by the conidia. Next one is which of the following are most suitable indicator of sulfur dioxide pollution in the environment? I hope everybody can answer this now. One is fungi, second lichen, third conifer, fourth algae. Very good. Everybody has answered so quickly. Very good, Veena. Very good, nursing. Very good, Srishni. Which of the following are most suitable indicator of sulfur dioxide pollution in the environment? Lichen. Remember, lichen is a very good pollution indicator. They won't grow in the polluted area. They are the symbiotic association between, very good, Shashank. Lichens, they are the symbiotic association between algae and fungi. Okay. Please don't get confused between a lichen and mycorrhiza, okay? Remember that lichen is a symbiotic association between algae and fungi. Mycorrhiza is a symbiotic association between fungi and the roots of higher plants. Okay, example, pinus. Now, question number 6 is pick up the wrong statement. 1 is nuclear membrane is present in MONERA. 2 cell wall is absent in Animalia. 3rd is Protista have photosynthetic and heterotrophic mode of nutrition. 4th is some fungi are edible. Pick up the wrong statement. Very good, Shishni. Very good, Veena. Very good, nursing. They're asking to pick the incorrect statement or the wrong statement. 1, nuclear membrane is present in MONERA. No, nuclear membrane is absent in MONERA because MONERA, they includes bacteria, and the bacteria, they don't have a well-defined nucleus. There's the absence of nuclear membrane. Cell wall is absent in Animalia, yes. Protista have photosynthetic and heterotrophic mode of nutrition, yes. The diatoms, the lignioids, they all are photosynthetic, right? Heterotrophic like climb mold, the protozoans, they're heterotrophic. Some fungi are edible, yes. Remember this, the truffles and morels, they are edible fungi. Please remember this. Nursing, please remember the morels and truffles which belong to Ascomycetes. They are the edible fungi. So the question is asking to pick up the wrong statement. So 1 is the wrong statement. Next one is mad cow disease in cattle and C.R.Jacob disease in humans are due to infection by 1 bacteria, 2nd virus, 3rd viroid, 4th prion. Nursing, that I have to check, okay? Very good, Shishni. Very good, Veena. So the mad cow disease in cattle and C.R.Jacob disease in humans is because of prions. Remember this. They contain abnormally folded proteins. Viroid, they causes a potato tuber spindle disease. Remember, viroid, they does not have protein coat. They have RNA as the genetic material, which is low molecular weight. Okay, and viroids, they are smaller than viruses. Remember this. And the viruses, they have the protein coat. They have the genetic material. Okay, and the virus, they cause a disease like tobacco mosaic disease. They can cause smallpox. They could be an influenza virus, right? So the different type of virus. And the question is asking about the mad cow disease in cattle and the C.R.Jacob disease. These are the neurological disorder which are caused by prions. Remember this. Next is which of the following statement is incorrect? Number 1 is viroids lack a protein coat. 2 is viruses are obligate parasite. 3 is infective constituent in viruses is the protein coat. 4th is prion consists of abnormally folded protein. They're asking the incorrect. Very good, Veena. Nursing, just now I've told. I've told the characteristics of viroid in the previous question. So the question is asking which of the following statement is incorrect. Viroids, they lack protein coat. It is true. The viroids, they don't have a protein coat. They only have RNA as the genetic material. Okay? Remember this. Viruses are obligate parasite. Correct. Infective constituent in virus is the protein coat. No, it's the genetic material. I told you. Genetic material is infectious. Okay? Prions consist of abnormally folded protein. Yes. So the question is asking about the which of the following statement is incorrect. Incorrect statement is number 3. I hope it is clear to everyone. I hope it is clear to you, Sushant and Nursing. So thank you all for your participation. We are winding up the class. I hope this lecture was informative for you. Thank you all and have a nice day.